if we look at, I want to look at how Jeanson calculated or looked when he did his pedigree mutation rate. He used a rate from a paper called Ding et al. Right?
Right.
And that was a rate that was calculated between parent and offspring. So from mitochondrial DNA, which is inherited only from the mother, that was the number of differences between the mother and her child due to mutation. He used a study from 2015 that compared mitochondrial DNA from between parent and offspring that was extracted from their blood samples, right?
Right.
Okay. And blood, of course, is a somatic cell. It's a working part of the body rather than a germline cell, right?
Right.
Okay. So it's a cell that it's not a cell that would be used to transmit genetic information to subsequent generations, right?
If it's a somatic cell
It's a blood
mutation, no.
cell.
Right.
Okay. And somatic cells divide throughout the life of a person in order to maintain homeostasis, right?
Yes.
Okay. And as those cells divide, they're going to start to accumulate mutations of their own, aren't they?
Of course.
Okay. But that's not true of a woman's eggs, which she's actually born with, right?
Yeah.
Okay. So if you measure the differences between somatic cells in an adult and their offspring, a bunch of those changes are going to be changes associated with mutations that occurred during that person's lifetime that are not going to be passed on to the next generation, right?
Right.
Okay. In fact, they'll be from three sources. You're going to find mutations from three sources. One, germline mutations. Two, somatic mutations that occurred in the parent. Three, somatic mutations that occurred in the child, right?
Right.
And those differences from numbers 2 and 3 could in fact be significantly more than the amount of change that you're going to observe in the germline, right?
It could be a higher mutation. So the mutation rate that was derived from the Ding paper could be higher than it really is because of these factors.
Right. In fact, it could be significantly higher, right?
Of course, if it's counting somatic cell line mutations, then yeah.
Yeah. And the Ding 2015 study that Jeanson used when he was calculating his mitochondrial Eve pedigree mutation rate included all three of those things, didn't it?
Right. I debated Dr. Dan Stern-Cardinal on the same topic 2.5 years ago.
Yeah.
You're not telling me anything I don't know.
Yeah, I remember before I told you guys that there were some things that he just misunderstands, and there are some things that he does understand, but he makes the argument anyways. Yeah, this will be one of those things.
All right. The only way that Ding gets you to 6,000 is because it includes a significant number of somatic mutations, right?
Here's the Parsons one. I don't look to the Ding one. So here's the Parsons paper.
So you don't use that pedigree mutation rate.
Right. Yeah,
Mm-hmm.
I agree.
Now that little mumbled concession may not have seemed like much, but that was actually quite significant because the graph that we've been staring at through most of this debate is taken from a book by a guy named Nathaniel Jeanson, and it is based on that study. And that's the ideas that Donny has been defending for years now at this point. And it is the only study that's going to get you to a 6,000 year rate. And as we shall see, neither the Parsons study nor any of the other studies are even going to get you close, which is why Jeanson used that study, why he misrepresented that study. And Donny's just admitted that you can't use that study. So that's actually quite significant.